discussions with reference genomes, and once we want to move a bit farther and start looking at, alignment and reference genomes and these concepts, something to put in a way to visualize the problem is think about solving a puzzle. Okay, think about solving a puzzle. So the jigsaw puzzle as an exercise for us, this is going to be it's always a very nice analogy to try to imagine the whole problem of sequence alignment. So we have here these pieces, right? We're gonna try to build the picture again. And usually, if you ever had, if you ever played this game, you would have a reference photo. Sometimes it could be small, right? I met some people who'd be really doing wonderful work, not just a small game. It would be hundreds and sometimes hundreds of pieces. Try to get a big picture of a nice drawing and so on and so forth. And sometimes it's small, you get for the kids at home as a gift and try to see if they can build. Always there is a reference, right? There's always a reference photo that they can check or image. So a strategy is to start with the edges, find the edges, and here you go, you are able to build all edges and then compare to the original picture, right? That is our reference. Think about reference genome. And so we have the picture now. We start identifying the colors and patterns. Now, this is one of the things that we as humans, when we do it, it's very straightforward for us. When it comes to computers, we need to find a good method, good algorithm, good way to do it, okay, to guide them into recognizing what is a pattern, okay, or what is a reference, and how to map all of these together. So we keep going with this nice animation example, and so we expand the with informative pieces. So we keep going, and then we keep going until we fill everything, and that's it. We get our image, which is for nice area for walking in some place. So in the central dogma again, we're looking to do this alignment to understand all with the reference genome to know what we are doing, because once we get some read, I have this sequence, okay, I don't know. Okay, I don't know what gene this read belongs to. What gene? Okay, so this mapping is certainly of interest to try to quantify the expression of that specific gene. So let's look at an example. Even if we have a bacteria or other examples, and again, we extract the sample, get the DNA, do all the fragmentation using the technology, go to our computer software, and now we want to do an alignment. Now, what I want to get your attention to is something that we are faced as an issue in this alignment. Okay, we don't have perfect alignment in this case, so look into that. So you see this, we're able to align these reads on top of each other as the animation kept going, and then we recognize some issues as compared to the previous example that we have looked at. Well, everything would seem nice. But here, what do we have? Well, to be able to align this read to this area of the genome, okay, we're going to miss some places. This C, for example, T and G here, and so on and so forth. So we end up with some areas that does not match to the reference. So alignment in this case is said to be strained, and generally this indicates one of the challenges or some of the challenges we face during alignment. When there is significant difference between the sample, that is the query sequence, okay, and the reference genome. If that difference is significant, then that is certainly referring to some difficulty in correctly mapping the reads from a reference genome to a reference genome. Okay, you know why we may have this. Why we may have this? Okay, because someone can come say, "Oh, Prof, why do we have this? Like, we got these reads from human. This is a genome, the reference genome of the human. Why we end up with this? Doesn't make sense, Prof. Okay, we need to have all nucleotides. They came from the same source, and they were mapping them back to the same source. So bear with me, and this is a very good question, and I'm gonna take this question and send it back as a treasure hunt question. So why do you think this may, why would this may happen? Okay, so share with me your answer on this part. Why would you think that this may happen? Okay, and then I'm going to give you some interesting facts, and so why we need to figure or consider this. Okay, another thing, if we're looking at a population that has diverse considerably at that population, so there is a specific population, and this may relate to one reason, good reasons. There's this population, this community that I'm dealing with, but they have diverged from the original reference genome. In that case, there might be numerous genetic differences, okay? And they can also manifest as single nucleotide polymorphisms, insertions, deletions, or even larger structural variations. Another thing is that genome often contains repetitive sequences, the genome itself. So if the sample has variations within or adjacent to these repetitive regions that are not present in the reference, okay, it can further complicate alignment. Okay, so if these samples are having the variations, the samples that we are working with and taking reads from, and these variations are, you know, affecting that alignment to these specific repetitive regions, and we're not able to map them to these regions, repetitive regions, we'll end up with some complicated alignment, okay? Some complicated alignment, okay? So let's go back, try to solve our puzzle, and then highlight a story where we may have this, okay? So this is one of the cases, and by the way, we may also have errors in the reference. So the reference genome may have some errors. It could happen, okay, depending on the case. So that would lead to misalignments or non-alignments. The story I want to highlight here is using common reference genome for all populations can introduce bias in downstream analysis. Okay, so that the traditional reference genome, that's referred to as, oops, let me stop here, pause a little bit before building this whole thing. Okay, I'm not able to write until the animation finishes. Okay, fine. Okay, so we may have a reference bias. Now, let's look into this, okay? The traditional reference genome that is highlighted as from the Human Genome Project and so on and so forth, there's version 37 and 38, is based predominantly in European or Eurocentric samples. So this is a specific population of humans, which can introduce biases when analyzing non-European genomes. For example, there is the Han Chinese genome CN1, okay? Now, a study that constructed a genome reference from a Han Chinese individual, referred to as CN1, revealed over 5 million SNPs. okay, SNP variants that were not present in GRCH 37. So this shows what? We need to be careful, okay? Yes, we have a reference genome, but we may have what characterize our own population. That's why it's useful not to have. So when we hear about projects nowadays, like there's the 1 million human genome projects in the States, that's interesting, okay? That's perhaps going to cover for many populations or communities, hopefully, okay? But there is one, this is one of the bias. There's also what the Pan-African genomes. The African continent has the highest genetic diversity in the world. And so when aligning the Pan-African genomes to a European-centric reference, many truly common African variants might be misclassified as rare or novel, because this is a bias. You are using a reference genome. It's like you're using a photo which is not the original photo, okay? So we get a confused output when we're solving that puzzle. We get something different, okay? So reference bias, in terms of a reference bias in the context of genomics, it is this preferential alignments of sequence reads to certain regions or alleles in a reference genome, which is going to lead to incorrect or incomplete variant identification. And again, some of the references or causes for reference bias could be population representation. And if the reference genome is predominantly represents one population or individual, so one kind, okay? Sometimes it's structural difference, so large structural variations such as inversions, duplications, or deletions, which are present in the sample, but again, not in the reference, and so on and so forth. So now, as we look into this, we examined this before, that generally as humans, we expect that the difference between us is less than 1%, right? How much that is to be manifested when it comes to certain variations and diseases and phenotypes, this is a point of interest, because one may say it's less than 1%, but is that significant or not, is something we need to keep an eye at. One of the ideas used here, which is 1 SNP per 1,000 nucleotides, this measure here refers to the frequency, okay, to the frequency of one single nucleotide per morphism per 1,000 nucleotide, okay? So a SNP itself is a variation in a single nucleotide that occurs at a specific position in the genome. That is what a SNP, it's a variation in a single nucleotide. So only one nucleotide at a specific position in the genome. And so where different DNA sequences are found in different individuals or populations, or alleles even, the different versions of that DNA sequence. So for example, at a specific position in the human genome, the nucleotide might be adenine, okay, in some people, and cytosine in other people, okay, in some people, and this is another group of people at that specific position, at that specific position. And so we refer to this as SNP. So when we say there is 1 SNP per 1K nucleotides, it suggests that on average, for every 1,000 nucleotides in the sequence or genome being discussed, there is one position where a SNP occurs, okay? So on average, for every 1,000 nucleotides, for every window of 1,000 nucleotides in the sequence or the genome text that we have, there is one position where we have a SNP that would occur, okay, on average. So this can indicate the, the genetic variation in population. Like in the context for the human genome, the SNP rate is roughly 1 SNP per 1,000 nucleotide. And this translates to over 3 million SNPs in this case, over 3 million variants given the size of the human genome, okay? So this is two different ways to look into these variants. So for humans, we have about 1 SNP per 1 thousand nucleotides, we end up with 3 million SNPs, giving the size of the human genome. So this table gives a reference, and so you can look at it and go through it. You would see different types of variants, like structural variants. We looked at this before, and their importance: total SNPs, single nucleotide variants, 3 millions in human, and so on and so forth. So these kinds of dissimilarities, although they are, in terms of when we look at the human genome, they are not, it's just a small, very small difference, right? It's less than 1%, but this can be emphasized or boosted when we go to the downstream analysis. There, when we have a huge effect. In other words, when we go to the downstream analysis, we're going to focus maybe on a few genes, right? Maybe a few thousands of nucleotides, and that's our area of interest. And within that area of interest, we see these huge variations. Okay. Now, this is how, again, dissimilarity affect analysis. So if we have two samples from a treatment and a patient group, so we get reads that we align to the genome, okay, and then we have another individual, for example, it's the patient or the control reads. So we have two types. The blue and the green here are referring to two different individuals. Now, the first individual, we got the reads and we were able to align them to the reference genome, okay? Because of some individual differences, the second individual, we were not able to map all reads, okay? Many of the reads were already excluded. That is an issue. That is an issue because specific area of interest in that case would show, oh, the gene is expressed in this individual, not in this individual. This is because we discarded many of these reads. So this is when it becomes tricky, when we don't have a sufficiently representative reference genome. And again, for handling these issues, there are some solutions, okay? For example, aligner parameterization, okay? And aligner parameterization can be crucial when trying to maximize the accuracy. In this case, okay, this is more about adjusting parameters, okay. We may have challenging genomic regions, diverse samples as we had in this case. And so in this case, what we do, we try to play with the parameters, okay. For example, mismatch tolerance, when we are working, try to increase the tolerance of mismatches. For example, increasing the number of allowable mismatches. It could help in aligning reads that originate from regions of the genome with variants that are not present in the reference genome. Okay. And this also gets us into different individuals here. So this is more of an evaluation of the GRCH38 reference genome. And then there are, like in the beginning, 93% of this genome primarily is a primary assembly, primarily was assembled of sequences from 11 individuals, 11 clones, okay, clones. So RP11 in this case, RP11 refers to, or RPC1 refers to the human library, a back library, which has a higher representation than all others. And this contributes to 70% of the primary assembly. Now, again, PAC library is this bacteria artificial cloning for cloning and trying to sequence this information. Anyway, the donor of RB11 library, the person who donated that was an anonymous male. But analysis suggests that his DNA is more of an African European admixture. Okay. CDD in this case is East Asian. So if I go to the plot here, this is East Asian, this is East Asian. And so kind of looking here how based on the cloning proportions, how these genomes comes together, okay? And you can see, yes, depending on certain populations, there might be some differences. And again, the reference genome itself, keep this in mind, it is composed of a group of, eventually this was extended, and so 93% of that, again, was made of 11 individuals, okay, and clones. Nice. So there's no such thing in this area as the average person. Let's have an average person for everything. It's difficult, but again, we use average as a technique and to overcome some of the challenges. So I'm going to move on into genome indexing, which is this first step that we want to tackle. This is the first step that I want to tackle in terms of the whole alignment process, genome indexing. And this is going to be the focus of this module. Now, I think this is interesting to bring your attention to genome indexing and dive a little bit more into this specific step to highlight some concepts that we use in borrow from computer science and the, specifically algorithms, to how to design solutions. And this is the first step. So kind of gonna give you a flavor of, okay, if this is the first step, then generally what's gonna be next will also include some algorithms and details. And also we're gonna go over some sequence alignment. So what do we see here in this plot as referred to as we start genome indexing? And I want to remind you here that since the genome is very long, so if the alignment algorithm requires a search along the entire genome for every read, as I've explained earlier, this is going to be suboptimal. So we need an index. Think about, you know, when you get a book and you need a way to get the location of sorry, for specific a topic or specific story that you want to read. So here, what you see in this picture is an overview of read alignment algorithm. Okay, so in step A, we have the seeds from the reference genome sequence are extracted. So this is the genome sequence. We get the seeds, okay? And seeds, these are the short reads that we are extracted, and each extracted seed, okay, as well as its occurrence locations, the reference genome, are using the data structure of choice. There are many data structures, so this is what is referred to as a suffix tree. We're going to look into this in today's lecture. This is more of a linked list and hashing. So there are different ways into how we can store data in computers for efficient access. Now, why this may look fancy or something, you know, new, what I want you to see is that instead of going through one shot in a linear fashion, let's break this down into a tree structure. So instead of having to visit everything, we decide right at the beginning, oh, if this smaller, like than a specific value or higher, think about, for example, if I'm doing a number comparison, so this is 45 and this is 3, for example, and always I guarantee the right is greater than this value and always this left side is smaller. So when I come here, I know, okay, jump here, okay? This is kind of the tree concept, though suffix trees are different in the way they construct the tree. And so we're going to look into this, okay? But again, it's going to save me. And there was called hash table. So I know all the locations of one specific seed, and we're also going to study that. So this is where the indexing step starts, okay? And once the branches and the suffix trees or hash table or other data structure are used, we come to step C. So now the seeds are extracted from each read sequence. Okay, so we get seeds from the genome, we build our data structure, and then we go and get the seeds from read sequences. And next, the occurrences of each extracted seed in the reference genome are determined by querying the index database. Okay, so the occurrence of each extracted seed is determined by querying, seed querying and filtering. And in this step, what's happening in this step, for example, as we see here, there were 3 reads from the first read, okay, and they appear at the adjacent locations. So we have these 3 reads, they are mapped to here, okay, as we extract those ones. Okay, we're able to map them into locations 5, 7, and 9. So we accept that those are adjacent from the location list, okay? But some, as we use the reference here, some are distant, so we don't accept these, okay? In this case. So 2 of the same seeds appear adjacent at another, at another location also, which is 12 and 16 in this case. This is 12, this here, and there is 16 for this one, okay? So there is some adjacency, okay? And so next, the adjacent seeds are linked together to form a longer chain, okay? So these are the seeds here, and those are the adjacent ones using the reference. So these are the reads here, these are the reference. So we link those together to be able to form a longer chain of seeds. And we need to keep an eye on the mismatches and the gaps. Now, pre-alignment filters can also apply to quickly decide whether or not the further computations or calculations are needed. Okay, and finally, once the pre-alignment filters accept the alignment between the read and region of reference, we move to alignment verification. So let's dive more into this step here. So first, there's an exercise. Find all occurrences of CCT, GTCT, C. Okay, I'm going to give you a moment here. Think about it, try to figure it out, and then share with me your answer. Now, as part of the treasure hunt, I understand this might be tough, so you can't tell me that I couldn't find it. Okay, but I'm going to give you a moment. Okay, tell me how many did you find? Okay, and it might be interesting because taking our eyes through these repeated patterns is like decoding the actual treasure hunt box, treasure hunt secret message, which is giving us the location for the treasure. Wow, right? So it's not easy to read. So these are 3 locations here. These are the 3 occurrences. So is there a way to enhance this skim through and take this whole reference genome, and if then break it down into a very neat piece, then we make searching this much more efficient. Because in this case, we may need to go through the text for one read, okay? For one read, we have this 3, so this is a pattern of 8. So say k equals to 8, our k-mer or our pattern is of length 8. This is our genome text of length n, so we need to do n, the length of this genome text, minus k plus 1 comparisons. So we need to do n minus k plus 1 comparisons for every single read. It is a lot, okay? It is a lot, okay? And if we end up with, you know, billion characters for billion characters here, and for one sequence we end up doing billion comparisons generally, this is a lot. What you are hoping for, what if we can do it through one comparison? Isn't a big save in time and efforts? Of course. So if we had an index, for example, for 3 sequence letters, then we can directly jump into, well, I'm dealing with CCT here, so at least I can reduce that, right? So I have CCT, GTC, so then I can jump directly here and know, okay, this is the part here that I can expand, I move on. So although older alignment software such as BLASTblock can be used to map short reads to reference genome, those methods are simply too slow. There have been many methods in the domain and field of short read mapping. This is an inherited challenge, given that we have short reads. All of this discussion throughout this module is because we have short reads. If we had long reads, things are going to be different. Imagine if we have the full DNA in one shot as one long read, then we don't need all these algorithms. So I don't know, okay, but this might be one of the ideas into how quantum computing in the future may change this. Maybe we'll have more powerful machines. Maybe we have algorithms that work more efficiently. It's about redesigning some of the algorithms. So yeah, it might seem interesting how to look into this, how quantum computing could change this area of you know, alignment in the future. So using indexes to solve the read mapping problem. We want to open the book. We want to use the index of specific terms in that book and refer directly to the location from where that term is coming from. So if I say term genomics, I want to read the text about genomics and all the pages that had the genomics appeared in it. So this is my query read, and this is my reference book, target book. So from the FASTQ file, I want to map the target genome until eventually I get the specific, you know, mapping for that specific read to the, after I know the position to the genome of interest. So how can we do this? So again, through this rapid lookup for some of the terms. Now, before I dive into this, let me tell you that indexes are widely used. Okay, even when you look into information in Google, they have an indexing system. Now, it is a sophisticated system using many servers, but of course they are using a very efficient indexing system. First, this means they have to build an index. So Google is doing this, for example, they look into web pages online, many resources online, and every time you have a website, there is a process to go and link your website to Google, and then they will build, you know, update their index based on this information. So when you go and search or query in Google, they have already a pre-built index. That's why they take you directly to the right web page or give you results in very quickly. Okay. So anyway, these indices are widely used, very important. This idea of indices and accessing information. So let's build our first index, and I hope it's going to be, you know, sort of straightforward and simple in this case. So the first type of index that we will build is called the Kamer index. And Kamer here refers to substring from the genome text. Okay, so we have our genome here, and we're going to extract different patterns or substrings. So we're going to cut this into pieces. This would be our first piece, or our first window. And this is a Kamer. We call it Kamer, and K decides the length of that substring. And then can shift my window. This would be the second one, okay? Usually this is how we work with patterns. These are three. We work with overlapping patterns. You don't jump directly to here, okay? We go into this here. This is the one here. And then this is, we move it one position, and then we move it one position, and then we build our patterns. So we'll extract all k-mers. So this is our first k-mer of length 3, which is GTG in this case, and then we're going to store its location, 0 in this case. Next one, we have TGC. So we're going to store 1. So we start with index 0, 1, 2, 3. One may say, why do we start with 0? It's for the convention, because some programming languages, okay. Okay, and usually, yes, this is useful for specific programming languages like Python and so on and so forth. Anyway, because they use 0 indexing. So we move to the next one, GCG. It's at position 2, and then we do this for all other sequences. So 0, 1, 2, 3, and GTG. This is 4. Okay. And so on and so forth until we get all of this list. Okay. Now I'm going to step back and ask you, what is going to be the location for this GTG? Share with me in the chat box. So I want you to work it out as I've removed these numbers, and share with me your answer as part of the treasure hunt. So we keep going, and then we ended up with the list of k-mers indices with the locations here. Oops. Next, we sort by alphabet. This is what's called lexicographically ordering. So you would find this. This is CGT, and then GCG, and then GGG. So we do this sorting here. Okay. So this is now, we have built an index. Let's see how we can use it to map a certain read. Imagine that the first read we get from FASTQ file is TGGGG. Okay. Now remember, the index was built using the reference genome. It has nothing to do with the FASTQ file. I want you always to, because everything looks like ACG. I don't want you to be confused. There's FASTQ file, there's index, which is coming from the reference genome. Okay. Since our index is built using 3-mer, that is the first 3 nucleotides. Let's extract the first 3. So this is TGG. And now we search through our array. So since this is already ordered, okay, what about using the value in the middle? I don't need to sequentially go this and compare, go here and compare, go here and compare. I know this is ordered. So in other words, if I start in the middle, this is GTG. I know that T, alphabet T in the English alphabets, is going to come after G. So let me jump into the middle of this one, okay, to save some steps. And so I know it's here. And now it's TGC. I know that TGG is going to be an award that will come after, because G, the final G, comes after this C. So I keep going. Oh, finally get TGG at position 7. Wow. And only 4 comparisons, or 3 comparisons, 1, 2, 3 comparisons. I didn't need to do 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. I didn't need to do 12 comparisons, only 3 comparisons, 1, 2, and bingo, you got it, right? Because we arranged and ordered everything. Now we keep going. What we need to go back to the original sequence and do some step of verification, because our original sequence was TGG and another G. And so we do it here, and then we find a mapping. And so this is how we use this to find, for example, or index some of the information, and then do the sequence alignment using this index. Okay. So I'm going to stop here, and then we will continue in the coming part.